From the Babylonians to the Romans and to the World Wars, in our human history there has always been an arms race. Traditionally, an arms race is a competition between two or more countries to determine who has the most available and powerful weapons. Each nation strives to produce the most technically advanced and destructive weapons. But what is an arms race from a microbiological perspective? Well, in fact, an arms race can be used to explain the biological relationship between two or more microbes where one of them is harmed and the other comes out on top. In microbiology, this is known as antibiosis. It usually occurs through the production of a metabolite by the dominant microbe that either itself is detrimental to the other microbe or it causes changes in the environment that favors one microbe over the other. Two microorganisms will need to be close together to determine if there is an antibiosis interaction. In antibiosis, there is only one microorganism that gains while the other is harmed. In a military sense, one microbe has less powerful weaponry and therefore cannot protect itself and is reduced in number or destroyed. Now, in soil microbiology, antibiosis can actually play a fundamental role. Within the soil of tobacco crops, particular strains of the bacteria Actinomyces produce antibiosis activities towards species of storage and field fungi. Actinomyces species found within the soil are strongly antagonistic against these two fungi in the tobacco crops. What Actinomyces does is it utilizes its secretion of toxic antibiotic substances to poison the other fungi. This antibiosis activity is actually used during tobacco crop harvesting. The storage crops are sprayed with culture filtrates of Actinomyces to prevent mold growth. Apart from soil microbiology, antibiosis occurs even in the depths of the ocean. In actual fact, antibiosis is very common between marine bacteria, but it's the weapons or metabolites it uses that will determine who is the winner of a particular arms race. For example, a study has found that when the Altomonas species of marine bacteria interact with a wide range of other marine bacteria, they actually attack them by producing a metabolite called SWAT5, which from that creates a product known as PQ, which completely inhibits the growth of its enemies. It does this by directly inhibiting DNA synthesis, protein synthesis, and bacterial motility, thus having a huge effect on the marine bacterial communities it comes across. From our studies of antibiosis, we have actually discovered the antagonistic effect of some fungi. For example, the antibiosis between penicillium and some pathogenic bacteria. The growth of the pathogenic bacteria is inhibited by the presence of penicillin, the active compound of penicillium. It does this by inhibiting the enzyme responsible to form peptide bonds for bacterial cell wall synthesis and thus weakens the cell and lyses the bacteria. Many years of research has led to the isolation of especially productive penicillium strains for use in the mass production of the antibiotic. Today, penicillin is available as capsules, chewable tablets, and powder for injection, and widely used to treat and prevent a variety of bacterial infections for both human and animals. So, when we think of an arms race, we usually think of all our wars in history, but really they occur all around us in our environments. From the depths of the oceans to the fields that surround us, and even to the most inner parts of our bodies, microbes are constantly battling each other to survive. This antibiosis results in one or more microorganisms being killed or suppressed because it doesn't have enough weapons or metabolites to overcome its enemy, just like in our human war history.